Hello to all. We should start. Welcome to this new symposium of OENOVITI International Network. I am very happy to welcome all of you. I would like first to say that, of course, the format of the symposium this year is again in video, unfortunately, because of the pandemic that in Japan was not over before the time we were thinking it was possible to advance. But we should have really the real symposium in Japan next year. That's the most important point, and we hope that all this pandemic will be not anymore trouble. This is the first thing. I want to thank also our colleagues from Japan that were involved in all the event and meetings that we had for the organization. You know that this symposium is in fact on the topic on the resilience in the grape and wine industry. It's really something specific with differentiation concerning vine or viticultural approaches and enological approaches, economical approaches that will be in fact morning, afternoon, at least between 11 o'clock to 1 o'clock the morning, and afternoon between 2 to 4. So this is what I wanted to say. The second point is that, of course, you know that after the symposium, Wednesday, you will have, in fact, the organization of the General Assembly of the network and also the working group of the network to incubate new project. So thank you to all participants to be part of it. And so I have a special thinking to our colleagues from Ukraine that are, as you know, all of us actually with some difficulties. We hope that the situation will be better for them, and we welcome them here to the symposium and to the event very warmly, and we hope to be able to help them. So I will let the word to our two colleagues from Japan, to Torhu Okuda to welcome you first, and then after to Shigeaki Oda, and we will start the presentation. So Torhu.
Thank you very much. I'm Torhu Okuda from University of Yamanashi, and thank you for giving us such a great opportunity to introduce our wine industry and our challenges. Anyway, I think that to save the time, I will give my mic to Professor Oda. Okay.
I'm Shigeaki Oda. I'm a member of Pastoral Center in Medical Research in Kyoto. I'm very happy to meet you online. Now, Japanese session in next year, and now this symposium very I hope successful close, right?
Thank you, Torhu, for your nice welcome, and I think that we are in time, so we can probably begin the first talk. So the first speaker is is Torhu. He will present to us the Japanese wine industry with a resilience toward humid climate. So Torhu, you have the floor.
Okay, thank you very much. Now I will share my slide. Okay, thank you for waiting. And again, I'm Okuda from University of Yamanashi. Recently, the quality of Japanese wines has improved significantly. However, since the amount of production is very small, it is about 1 million hectoliter. I think that many people have never drank Japanese wines. Japan has a lot of rain and very humid, so it is not suitable environment for grape growing. In order to make grapes in this environment, many non-vinifera grapes are cultivated in Japan, and many wines are also produced from these varieties. I hear that non-vinifera grapes are currently attracting attention all over the world to cope with global warming because of their strong resistance for fungal diseases. Japan's long try to non-vinifera wines may be useful to everyone in the world. So first of all, I would like to talk about the current state of Japanese wine production, focusing on non-vinifera grapes. Okay, first let me explain a little about the history of grapes. It is said that the grape family appeared on the earth about 10 to 15 million years ago. Continental drift was not completed during this period, and it is believed that it was much warmer than it is now, and that there are no ice in the Arctic area. The grape families was widely distributed in the northern hemisphere. Then some glacial period came. The northern part of the earth was covered with snow and ice, and vines had no choice but to move south. Because of sea and high mountains, grapes survived and developed in three areas: Europe, North America, and Far East. Grapes that survived in Europe is named Vitis vinifera, and most of wines are made from these varieties. Grapes that survived in North America, there are some varieties. Labraska is very important for table grapes, and others are important for rootstocks for grape vines. On the other hand, grapes in Far East is not well known. Because Japan is located in Far East area, Vitis amurensis, Vitis quaniti, and Vitis fisphoria, and others are native to Japan since more than 10,000 years ago. Grape seeds about 6,000 years ago were excavated at the Sannai Maruyama site, and Japanese people may have been making wine since this time. However, there is no clear evidence. In the 8th centuries, Koshu was brought from Europe through China via Silk Road. On the way to Japan, Vitis vinifera met a wild grape named Vitis davidi and hybridized. This fact was revealed by DNA analysis. However, the father of Koshu is still unknown. About 150 years ago, the Japanese government decided to westernize the Japanese culture. the government forced the Japanese to quit the samurai, eat the same things as the West, wear the same clothes, and force the same culture. Government also introduced various grapes from overseas. Many vinifera from Europe, and Labraska was from United States. At this time, the Japanese were not interested in wines, so grapes were planted for eating. To breed delicious table grape varieties, many crossing have been carried out. Even today, 80% of the grapes planted in Japan are edible grapes, and unsold grapes were processed into wines. Because Japan is island country, grapes have continued to develop independently, and various varieties have been bred from various types of grapes. That is why the grapes in Japan are very, very diverse in their species and very complex. Phylloxera was also entered Japan, and it is known that it caused great damage to vinifera that came from Europe. On the other hand, Labraska, which came from United States, it became popular in Japan because it was resistant to both phylloxera and various diseases. And when Japanese people started to eat Western food, we became interested in delicious wines. And now wines are very popular in Japan. This is the grape varieties that's grown in Japan for winemaking. As you can see, Koshu is the most produced wines in Japan, and the next production is Muscat Berrier. In this table, red character shows Vitis vinifera, and blue one is Vitis labraska and its hybrid. Black character, Yamabudo, is Vitis quantity. I think you can understand the very, very unique varieties of Japanese wine grapes. The most famous wine producing area is Yamanashi. It is located in the middle of Japan. Koshu and Muscat Berrier are the main producing varieties. The climate is very hot and humid, especially in summer season. We used GDD, growing degree days, to classify the climate of Japan. Japan is long from north to south and has many mountains, so the climate is diverse. GDD calculates only temperature as variables. But for grapes, the amount of sunshine and rain are also important variables. The figure on the right shows the integrate degree daylight hours, red line, and rainfall, blue bars. In Japan, we have many rains during the growing season. Heavy rain leads to fungal diseases. Also, on rainy days, the hours of sunshine will be shorter. This is an example of last year, 2021. It can be seen that there is no sunshine for about a week due to rain here, here, and here. This can have a significant impact on grape growth, and sometimes accumulation of sugars completely stopped, even in a mature period. Details are still in the research stage. There are many non-vinifera grapes in Japan. They each have very different characters. Because most of wines are made from Vitis vinifera grapes, there is very limited knowledge about non-vinifera grapes and their wines. With a long effort on our research, some features of these grapes have been gradually elucidated. Vitis amurensis and Vitis quantity and their hybrid are very rich in anthocyanins. Sometimes the wines look like black ink. One of a Japanese researcher showed the content of anthocyanins. Can you see the Cabernet Sauvignon, the Merlot in this figure? These vinifera varieties contain monoglucosyl anthocyanins expressed as a 1G character here. However, Vitis amurensis and Vitis quantity and their hybrids has many 3,5-diglucosyl anthocyanins. In this figure, it expresses GG, double G. These double G anthocyanins may have tolerance toward oxidation. And on the other hand, some Labraska hybrids have low tannin content. Tannins are located in both skin and seed of grape berries. We revealed that Muscat berry have very low content of tannins. One reason is very low content of seed tannin in Muscat berry. We also showed that skin cell wall of Muscat berry has strong tannin absorbing capacity. Because of low content of tannins, Muscat berry wines are usually light in taste with low astringency. This character matches with Japanese cuisine, especially seasoned with soy sauce or miso. On the contrary, Koshu is known to rich in phenols as white wines. As shown in the figure, skin of Koshu contain many phenols. Many attempts have been done to improve the taste of Koshu. Surely method give the thickness on their taste. Some company employ hyperoxidation technique to reduce phenols. Recently, orange wine from Koshu is very popular. This method strengthens the taste of phenols. There are many types of Koshu wines exist now, and it is very good combination with Japanese cuisine, especially seasoned only with salt. For conclusion, there are many non-vinifera and their hybrid in Japan. Fungi tolerance is one of the most important issues in Japan, and Japanese wines are very good combination with Japanese cuisine. From this 150 years of Japanese wine history, we learned that each variety has very unique properties, and most of these properties are brought from non-vinifera grapes. We are currently updating our winemaking method daily to adapt these varieties. The Japan wine competition has been held since 2003, and the quality of wine has steeply improved. Today, I've focused on the story of non-vinifera wines, but there are also many great vinifera wines. Next year, the OENOVITI International will be held in Japan face-to-face. We really hope to meet all of you Japan in next year. Thank you for your attention.
Thank you very much, Professor Okuda, for this very nice introduction with a lot of information concerning varieties and the vine you have in your country. So maybe the proposal can be that we should listen all the presentation and have the question at the end, if everybody agree. So then we can, in this case, continue with the second speaker. The second speaker is Professor Enoki, who is talking about genetic characteristic of Japanese indigenous grape, Koshu, for high-end on white wines. So I don't know if Professor Enoki can. Yes, we can see him. So the floor is yours.
Hello, everyone. Thank you for your time today. My name is S.ENOKI. I'm a researcher in the Fruit Genetic Engineering Research Division at the University of Yamanashi. This time, I'll be speaking of the subject of genetic characteristic of Japanese indigenous grape Koshu for high-end white wines. As for my English, it is poor, but please forgive me. Then let's go. First of all, what is Koshu grape? Koshu is a grape cultivar indigenous to Japan. Koshu has been cultivated in Yamanashi Prefecture, located in central Japan, for more than 1,000 years. Here, at the region where Koshu is grown, the map of Japan where Koshu is grown. The map of Japan shows the yield of its region. It is widely grown in various regions, some of them such as Osaka, and most of them are grown in Yamanashi Prefecture, which has in this Mount Fuji. Here is a Koshu appearance. It looks like a Pinot Gris grape. A Koshu is the most important cultivar for white wine production in Japan. The common belief was the origin of Koshu is that it came from Caucasus region, proceeded China, and settled in Japan. The lineage was revealed by Goto Yamamoto et al. The Koshu lineage was the result of two crosses between wild Derby from China and Vitis vinifera from Europe. For this reason, a Koshu has many characteristics that distinguish it from vinifera. Here is one of them: the size of berries. This Koshu has larger berries than European vinifera grapes, such as Chardonnay and Cabernet Sauvignon, and similar berries than Zenkoji and Muscatberry which are similar to American grapes used for table grape. The most significant characteristic is still the light purple skin color, like this. Grape skins are classified as red or white based on the amount of anthocyanins in pigment they contain. The red grapes, Cabernet Sauvignon and Merlot, have a large amount of pigment, while the white grapes, Riesling and Chardonnay, have no pigment. In contrast, Koshu grape accumulate only small amount of pigment. So what is the reason for the low anthocyanins accumulation in the Koshu grape skins? Previous studies have shown that the cause is the structure of the MIB-A1 gene. MIB-A1 is a transcription factor that regulates pigment synthesis genes. The grape chromosomes in figure A has a white and red allele. If both they are white, pigment synthesis does not occur. The white type has a retrotransposon, a white one, in the MIB-A1 structure, whose expression is replaced. The white type, the MIB-A1 structure analysis of many varieties by PCR in figure C shows that a Koshu MIB-A1 structure is similar to that of red grapes. Furthermore, this MIB-A1 structure was investigated in different cultivars like this. Koshu has a red allele gene. Red allele of the MIB-A1 gene, but Koshu have a 33 base pair fragment in the second intron of the MIB-A1 gene in red allele. This short insertion reduces the transcription activity of the MIB-A1 gene. As a result, the color of the grape skin is weakened. Another important feature is the high polyphenolic content. The hydroxamic acid and monomeric flavonoid content in Koshu grape juice is higher than in the juice of other white Vitis vinifera cultivar like this. Koshu is higher fat content. On the other hand, Sauvignon Blanc and Chardonnay is a little. This gives Koshu a white wine as light astringency. Thus, Koshu has many characteristics other than its fruits. Koshu is vigorous to eat. For example, it is also grown in a uniquely Japanese shelf-style method called overhead trays. Research on Koshu has focused mainly on the analysis of component and cultivation method and the genetic relationship between characteristics and genetic relationship has been unknown. Therefore, the objective of this study is a characterization by a genome analysis. The whole genome sequence method was used to decode the Koshu genome. This is a method of mapping and reconstructing the decoded data of the Koshu by referring to the existing genome decoded reference cultivar Pinot Noir. This is here the result of preliminary phylogenetic analysis using the whole chloroplast genome. 3 cultivar was used. Koshu, Thompson seedless, Tana. Thompson seedless, white, 3-way used grapes, wine, table grape, and raisins. Tana is a red wine. Koshu is an object. Therefore, we decided to enter genome and to analyze the genetic factor controlling the characteristics of Koshu. First, here are the features of the whole genome structure. Here are details. The ratio of homozygous cells and heterozygous cells, variation of chromosomes 1 to 90. SNPs, single nucleotide polymorphism were compared to among these 3 cultivars. Only Koshu was characterized by high number of heterozygous mutation in chromosome 7 like this. This will help in the development of genetic marker to identify to Koshu. Next, what change do the mutation gene causes? We compare the amount of mutation accumulation biological processes and metabolic and synthetic pathways required for survival. From left to right, Koshu, Thompson seedless, and Tana, with a red indicating higher mutation levels. In BP biological process, only Koshu has mutation in processes relationship to stress responses. For example, response to water deprivation, oxidative stress, and plant type cell wall organization related to cell protection and own growth. These mutations are considered to be accumulated in favor of growth and environmental adaptability due to Koshu's strong vigor characteristic. On the other hand, no mapping between these pathways and characteristics was possible for Koshu-specific metabolic pathways. However, these are individual interesting metabolic pathways. This is a phenol related pathway related to astringency in Koshu. A comparison of mutation accumulation in the 3 cultivar of the phenylpropanol pathway, which produces phenolic compound, is shown in this figure. This pathway, the box is divided into 3 segments from left to right, Koshu, Thompson seedless, Tana. The lighter the box, the higher the mutation level. Interestingly, Koshu showed a similar tendency like this. Koshu shows similar tendency to accumulate mutation than Thompson series in white grapes and Tana in red grapes, which contains 6 times more polyphenols than Pinot Noir. Similarly, Koshu and Tana accumulation was similar in the metabolism phenolic compound in the flavonoid biosynthetic pathway, such as caffeic acid synthesis. The figure on the right showed gene expression level for this pathway. Here is a comparison of Koshu between and Pinot Noir of the difference variety. The more oranges, the higher the expression level of Koshu, and the more blue, the higher the expression level of Pinot Noir. The amount of mutation accumulation in this study generally match the amount of the gene expression in our own previous study. Here, as a similarity with the previous studies, the table shows the amount of phenol in each cultivar. Koshu actually has more phenylpropanoid and flavonoids than European red and white grapes, like Sauvignon Blanc, Chardonnay, and Merlot. Koshu, this is related to actual amount of substances and mutation accumulation in this study. The other focus was on the aroma component 3MH, 3-mercaptobhexanol. This is a citrus-like ingredient famous in Sauvignon Blanc and other products, and has actually been applied to the production of Koshu wines. An example is Chateau Merchant's Koshu Aroma project. This company has found a way to increase the volume of the 3MH component of the Koshu, and it has been well received. In collaboration with them, this is a metabolic pathway of 3MH that has been proposed so far. In these berries, it is synthesized from 2 substrates, namely hexanol and glutathione. To the more 3MH precursor of the various enzymes. In response, we focus on the 2 pathways that produce 2 substrates of 3MH. It's the alpha-linolenic acid metabolic pathways and the glutathione pathways. Here, the alpha-linolenic acid pathway generates hexanol production. Of the note is the junction black circle in the middle of the pathway. Here, there is HPL1 enzyme that produces hexanol. And the 4.2.1.92, namely AOS enzyme in this box, that produces a plant hormone, jasmonic acid. In Koshu, there is a high accumulation of the mutation in the gene encoding the AOS enzyme for downward JA synthesis. On the other hand, mutation in Koshu involved in glutathione production are scattered. The relationship between them is ambiguous, but so far the glutathione contained in Koshu is high, which is consistent with the gene expression levels in our previous studies. While we have characterized the genome in this study, future study will need to investigate in depth the clear correspondence between these variations and traits. Finally, here is a new 3MH synthesis hypothesis for Koshu based on our result. In Koshu, the amount of 3 production may be hexanol dependent, since glutathione is more abundant of the 2 substrates in the fruits, and hexanol is less abundant. In this alpha-linolenic acid pathway, A JA formation, a jasmonic acid formation may be a preferential from mutation accumulation at the substrate branching point. Based on the above one, hypothesis and ideal to destroy some of the JA biosynthesis gene by genome editing or other method. In this case, it may lead to applied research such as increasing 3-MH production of Koshu. In summary, in this point, Koshu genome information is expected to make a significant contribution to improving the taste and aroma quality of the Koshu wine. That concludes my presentation. Thank you for your attention.
Thank you very much, Professor Enoki, for your very interesting talk. And so we will take also the question at the end of the session. So the next speaker will be Professor ISHIDAIRA. Professor ISHIDAIRA will talk about global warming and land suitability for vineyard in Yamanashi. I just remember you before the talk of Professor ISHIDAIRA that you can ask questions in the little icons that you have just under the screen where you have a question and answer. Voilà. Feel free to answer question. And so, of course, we will take all of them at the end of the session. So, Professor ISHIDAIRA, you can have the floor. Thank you very much.
Okay, thank you very much. So I'm Hiroshi ISHIDAIRA from University of Yamanashi. Again, thank you very much for giving me an opportunity to have a presentation in OENOVITI International Symposium today. Actually, I have been worked in the field of the hydrology and water resources, not in the enology of the viticulture. I'm a very beginner in this field. However, very luckily, I had a chance to work with Professor Okuda and Professor Kishimoto in the Institute of Enology and Viticulture in the University of Yamanashi. So today, I'd like to introduce some part of the result from our collaborative works. So this is introduction. The climate change associated with global warming is anticipated to negatively impact wine grape production. So the assessment of the impact of climate change on land suitability for vineyards is indispensable for the sustainable development of wine industry, as well as for the planning and implementing the climate change mitigation measures. The several studies, as you know, have been reported distinct changes in the land suitability for the vineyard under the global warming at both the regional and the global scale, actually. However, most of the previous studies were conducted in Europe, US, and Australia, and few studies have been investigated wine grapes in Japan, especially in Yamanashi Prefecture. So the objective of this study is to construct a land suitability map for vineyards in Yamanashi Prefecture, Japan, that using the growing season temperature, shortly GST, as an indicator of land suitability, as well as a high-resolution gridded air temperature dataset in the past, present, and future. And another objective is to investigate the impact of the mitigation measures on the future land suitability for vineyard. So this is a study area. The study area is located in Yamanashi Prefecture, central part of Japan, and which has the area of 4,465 square kilometers and the population of approximately around 820,000. The study area is surrounded by mountains, including Mount Fuji and the South Alps, and also contains the Torhu Basin, which has altitude of around 200 to 300 meters at its center. Grapes have been cultivated in Yamanashi Prefecture for hundreds of years, and grape growing area scatters throughout the hilly area of this region. And these regions are remaining the grape production center of Japan, where a wide variety of grapes cultivated and wines also produced. Next, I would like to move on to the methodology part. So first is about land suitability assessment. In this study, the growing season temperature, shortly GST, is used for the land suitability assessment. GST is defined as average of air temperature during the wine grape growing season in this study area. Here we use define this the period as April to October. The range of suitable GST differs among grape variety, as you know, and also as shown in this figure, like this. And in the GST, if the GST on a certain place or area is within the range of suitable GST for the grape variety, in that case, the area is considered as a suitable land for that grape variety. That's a very simple approach. And also, we have used several climate data sets. So the high resolution gridded air temperature climatology data were used to determine the spatial distribution of GST to assess the land suitability for the vineyard, where the climatology means that the average of the typically 20 to 30 years of the climate data. Anyway, we apply the mesh climatology data dataset provided by Japan Meteorological Agency to analyze past to the present climate, like the 2000, 2010, and 2020. And also, future climatology data were obtained from World Clim, and these data consists of the multi-climate model, I mean, and we call GCMs, multi-GCM and bias corrected, and spatially downscaled future climate projections. Also, the output from the 19 climate model under the RCP 4.5 of the IPCC AR5, this is one of the climate change scenario, were used in this analysis. Then these data contain the monthly climatology of the air temperature, maximum and minimum temperature, with a spatial resolution of approximately 1 kilometer. Then I'd move on to the result part. As you can see, this is a land suitability map under the past and the present climate conditions are shown in this slide. Here, the map for the Muscat Perrier and the Koshu grape variety, as already introduced by the Professor Okuda and Dr. S.ENOKI. These are the major cultivar in this region, as shown in the slide as an example. In this map, if the GST for the grid cell was within a suitable GST range for the given grape variety, then suitability of that grid cell was scored 1, and for that variety, and otherwise it was scored as 0. So it means that this colored area is a suitable land for each grape variety. As you can see, the spatial pattern of land suitability changed even within the past decades from 2000 to 2010 or 2010 to 2020 in a very short period of time. Of course, although the significance and the trend of these changes differ among grape variety, that the area deemed suitable for vineyard effectively shifted from the lower altitude to the higher altitude area, means from the center of the valley into the higher mountains area slightly. And in addition, as you see, this is a slide, the graph, the total area of suitable lands also be changed in the past decades, like this. And the next slide shows the change of land suitability from current, the present to the future. A similar procedure, the process was applied for the average climate projection of 19 climate model output to obtain land suitability map under future climate, like this. From this is a present situation, and the 30 years later, in the 2050 in the future. In addition, the climate projection from each of the 90 climate models were also used to construct 19 different future land suitability maps. And then the overlay these maps allows us to create a unified map showing the probability distribution, the suitable land for the vineyard, like this figure. As you see, some gradation you can see in this map. From this result, we can say that the change of spatial pattern around suitability became more significant under the projected future climate condition rather than the past change. Maybe you can compare with the past, the previous slide, a more significant we can see. However, it is noted that the future projections involved large uncertainty, as shown in this map. You can see that even some parts, it's very low possibility, but they still have some chance to be a suitable land. Furthermore, the significant trend of these changes is different for among grape varieties like this. Some varieties are getting the areas getting down, but some ones the increase. In the previous slide, change of land suitability was represented in a form of a map. However, I like to show these change in a bit different way as well. So this graph showing the transition, the average elevation of suitable land for vineyard for the past, present, and future. Means that where the average elevation over the suitable land was calculated from the land suitability map combined with digital elevation map. See, this is the graph showing the transition of the average elevation. The detailed pattern of change of the trend is slightly different for each grape variety. However, the suitable land for vineyard have been shifting from the lower elevation to the higher elevation zone in the past decades, and also expected to be rise more about 200 meters around in the next 30 years. This slide is also showing the transition of suitable land for the vineyard, also in a bit different way. See, in this map, you can see some red point. This is a location of the major winery of this region. And the distance from each winery to nearest neighbor suitable land was calculated, and then averaged over all wineries. Then these graphs are showing the transition of average distance from winery to nearest neighbor suitable land for different grape varieties. So the significance of change are largely different among the grape variety, as you see, and also, but around the sum of the variety for the 15% of the increase was somewhat more than in double. But in general, the suitable land may move a little further away from the location of the winery and the changing climate. Okay, so in the previous slides, we could see the potential change of land suitability caused by the global warming. But the next question is how we can mitigate the impact of climate change. Actually, several measures have been proposed to mitigate the impact of climate change on viticulture by maintaining wine grape production, even at a high air temperature condition, such as the cultivar modification or the vineyard relocation. But the one promising approach involves the shifting the growing season by applying the specific cultivation techniques, such as the lateral shoot grape cultivation. As reported in Kishimoto and Yamamoto 2020, this cultivation method making it possible to delay the growing season by approximately 1 month in this region. And the shifting the growing period also provides a cooler temperature condition during the ripening and also be an effective method for improving the fruit quality. So we examine the impact of lateral shoot grape cultivation method on land suitability change under the future climate using land suitability maps produced in this study. And here we assume the 1 month delay in this growing season. This slide shows the comparison of the future suitability map for with and without the mitigation measure. Maybe, I'm sorry, this map is very small and very difficult to see the major difference between two cases. However, if we see in detail, we could find that the total area of the suitable land for vineyard has changed a very little, not much actually. However, some area of the suitable land will be remained to lower elevation zone, even in a future climate condition. So we can say that the shifting the growing season would work to mitigate transition of suitable land caused by the climate change in the future. And these graphs are also showing the impact of mitigation measure on climate change of the suitable land in the future. Each graph showing the changes in the total area, these upper panels, and the average elevation of the suitable land in the middle panels, and the distance to the winery for the suitable land at the lower panels for each grape varieties like the MBL or the Koshu or some others. And and you can see blue and red line representing the cases of with and without mitigation measures, respectively. Means that, so please look at the difference between these two lines, like red line without mitigation measure and blue line with mitigation measure, to see the impact of the mitigation measure on the transition of suitable land. So this time is limited, so I like to skip in detail, but I like to say although the significance of the impact is different for each group varieties, but shifting the growing season may be effective to mitigate the reduction of total area of suitable land, like shown in this upper panel. And also the effective to maintain the elevation zone for the suitable land lower, like middle panel. And also the effective for remaining the closer distance between winery and the suitable land. Okay, so I'd like to conclude my talk. In this study, the changes in the land suitability due to climate change were investigated using the high resolution climate dataset and suitable GST range for the various grape varieties in Yamanashi prefecture. Then, and land suitability patterns change between past and present climate condition, and also the change will be more significant under future global warming. And also the degree and trend of the changes in land suitability is greatly differed by the grape variety. And third one is the shifting the growing season, for example, by applying the lateral shoot grape cultivation, may mitigate the transition of suitable land for the vineyard to some extent. Okay, that is my talk. Thank you very much for your kind attention.
So thank you very much, Professor ISHIDAIRA, for also this interesting talk. And so we can, I think, now begin the discussion. It was one question that was asked already by our colleague Daizan Zhu. The question was concerning how the mutation accumulation was quantified or measured, and Professor Enoki was answering that it was difficult to explain, but after decoding the genome, non-synonymous substitution, mutation accumulation were detected and statistically processed using a specific tools that it was sent in a link from a Frontier in an article in Frontier. So you can be free to ask questions. Maybe I have one first question for Professor Okuda. The overview was very interesting concerning the history and the evolution in your country, in Japan. Do you think that there is what can be the future for some hybrid maybe in Japan? Do you have some idea about it?
Thank you for your question. It is very difficult to answer the question, but now, as I talked, the quality of wines from the hybrid is very, very diverse and different from the vinifera wines. So someone said that we need to cross back to the vinifera more than one time or two times, or then we can get a good quality of grape varieties. It is one idea.
Okay, but is evolution of climate favorable for that?
Yes, it's so. I think for the northern area, the Vitis amurensis and others are very good genetic source for cold tolerance. However, there are no high temperature tolerance grape varieties now, so that's a problem for me or for Japan, I think.
Okay, okay. Thank you very much. There is some other question. Alors, there is one question for Professor ISHIDAIRA from Zanzu Dai. Zanzu is asking, has the GST used in this study been refined for the local viticulture zone in comparison with those original ones from Jones? And in addition, could you please comment on the risk of spring frost in sustainability evaluation? Thanks.
Okay, thank you very much. Yeah, I agree with this idea. Actually, the range of the GSTs actually the somehow depend on the local condition there. So, but in this case, I just use the definition from Jones, and also for the Koshu and the Muscat Berrier, our groups identified my own the range based on the experiment, not based, but the literature review. But I understand that the definition of the GST for the suitable range is very, how can I say, not easy, but this case, just use the literature value. And also the next question is about the spring frozen. Okay, that is also very interesting, the topic. And actually, I didn't analyze this one, but checking the minimum temperature at this season will give some of the insight how it will be affect the impact to the sustainability. Thank you.
Okay, so this was for the first one. There is a second one from our colleague Professor Enrico Peterlunger from Udine. He's asking you, Professor ISHIDAIRA, as to the production on lateral shoots, this is dependent strongly on genotype. On
exactly.
This was experimented.
Okay. So I myself, but according to the previous report by Kishimoto and Yamamoto, for example, the Chardonnay, Riesling, the Semillon, the Pinot Gris, Pinot Noir, and Shiraz, this type of the varieties are tested for the lateral shoot, the cultivation. Of course, they are continuing to investigate other variety as well, I suppose.
Okay, thank you. I have one question for you also, Professor ISHIDAIRA. It's concerning the rain, because we have seen the temperature, we have seen the elevation, and you just answered concerning the frost. But what about the expected rain evolution? It should be more, it should be less. Is there some prediction there? So also for the future.
Okay, thank you very much. Yeah, that's a very good question. And also especially in the most of the area in Japan, the global warming will be contribute to the increase of the precipitation rainfall actually. But I checked this region. It's the prediction of the precipitation change is highly uncertain. Some model expected they predict the very much increase. Some are less or the decrease. But take the average. I checked maybe not much change in the future actually. So that is the situation. But again, the temperature prediction is rather, I can say it's not easier, but they are much easier than others. But the prediction of the precipitation change is much difficult and uncertain. That's why at this moment a bit difficult to say. But according to my analysis, not change much is expected.
Okay, thank you. Thank you very much. There is another question for you, Professor ISHIDAIRA. It's concerning how economically advantageous is the dislocation of current growing areas as compared to the lateral shoot growing in the current area. From Antonio Graca, from our colleague from SOGAP.
Yeah, okay, thank you very much. It's also very interesting and very important question, but at this moment, I have not clear answer. But the reason why I made this type of map is if you like to estimate the necessary cost to be relocated or some other options, this type of map is very useful because we can identify the potential area and how far from the winery or some sort of the things we can calculate. Then maybe next step is to quantify this, which option will be, how much cost will be necessary, those sort of things. So maybe this is the next task and I hope to report in the next time.
Yeah, yes. Not easy to fix everything.
Exactly, yeah.
Yes. I have maybe 1-2 other points that I was thinking. The first one is concerning the Koshu, and maybe also for Professor S.ENOKI, maybe also for, well, all of you. Maybe I don't have enough culture, but I don't know. So Koshu was introduced finally at the beginning for table grapes or for wine purpose first. What was in fact the first goal of the Koshu? It was table grape or it was to do wine?
It should be answered from the Professor Okuda, I suppose.
Okay. Actually, Japanese people drink wine, did not drink wines because we have sake. That is from rice. So Koshu was used for table grapes for a long time.
Okay, for a long time, and then finally it was moving to the wine, and so this is very interesting because do you have a lot of different clones of Koshu finally, or not, and how it's working in terms of when you are doing the plantation? Do you have a large clone possibility, diversity, or is there a few, in fact? So that's also one of the points, maybe.
Now some researchers studying about the clones of Koshu. However, it is very difficult. I think there are some clones, and actually there is some vines have very big berries, and others are very small. However, I think there are no data for, especially for the genetic data for such kind of clones.
Okay. And so the yield in this case can be a limitation factor in terms of I don't know, aroma or aroma precursor or phenol. Do you think that the yield is a very important issue for the Koshu?
This is for Dr. S.ENOKI? No?
Yeah, it can be. I don't know if Professor S.ENOKI is there. Or you can answer if you want to.
Oh, okay. It's very difficult to say. And yes, actually, some clones of Koshu has very different smell, and I think there are some possibility, but I'm sorry, I can't answer that question.
Okay.
I'm sorry.
No trouble. There is another question here from always our colleague from Portugal, Antonio Graca. So he's asking for all of you, overall, how do you see the future of Japanese wine? It's very foreign variety brought in, more local variety bred to adapt to climate change, new area being used for growing grapes. So how will be this future?
Thank you very much. Now we are trying to import the European varieties because especially from the Spain or Portugal or something, because it is good for the global warming, and that is why we are trying to import the seedlings. It's very difficult to say, but some many consumers want to drink the vinifera wines. That is problem for us, and others are very interested in the grapes native to Japan. For example, Vitis amurensis and Vitis coaniti. There are the two types of consumers. So that is why it is very difficult to predict the future status of Japan.
Okay. So if there is two possibilities there. And maybe Professor ISHIDAIRA, what do you think?
Well, it's also a bit difficult question, but anyway, no single solution we could have, but the several types of the approach or the countermeasure need to think about. Of course, brought the varieties also be one of the options. Also but anyway, several kind of the options need to be think about. That's I'm thinking.
Okay, thank you. One question also maybe is concerning a point that is also very important there. It's a disease. And so we know that because of the climate evolution and the actual climate you have in Japan, some disease can be there and can be sometimes trouble. So do you have Koshu with disease resistance work, or did you try some maybe some possibilities there to have something more resistant to disease? Can be for for all of you.
I'm sorry, I'm not a viticulturist, so it is very difficult to answer the question.
Okay.
But as far as I know, some of the researchers or professors in the Institute of the Enology and Viticulture in the Okuda Sensei's Institute, some others are doing this type of work. And so next time maybe we can ask him to have some presentation.
Okay. You have another question concerning for Professor Okuda. It's Enrico Peterlunger. He's asking how difficult is grapevine defense in Japan against downy and powdery mildew. How many pesticide applications are needed, in fact? So, well, it joins a little bit the past question, but yeah, it's very interesting point also. What is your approach there?
Regarding to the fungicide, I'm not sure that I think we spray the chemicals more than 20 times in a year, and one of the problem is acquisition of tolerance to other chemicals. So now many people try to change the chemicals so often to prevent the acquisition of chemicals, tolerance to other chemicals. And is this answer for you?
Yeah. So it means that actually you have a number of treatments that looks enough important, and so. But is it an objective to decrease the number of treatment, in fact, for the future, or do you have some? Well, you have probably some research program on this point, no?
And I would like to explain one more thing. The problem in Japan is not only powdery mildew and the most important problem is ripe rot. Some chemicals are very effective for the powdery mildew and for example, botrytis. However, now it is very difficult to prevent the ripe rot. That is a problem for us.
Okay. So you have well, some typical diseases that are also different of the traditional ones that we know, in fact. So that's also a very important point here. Okay, and another point, maybe, I don't know if Professor Enoki is there, but he was showing the level of anthocyanins in Koshu, and also the fact that there is a glutathione or a possibility of genesis of glutathione. So the level of glutathione is high in Koshu in comparison with some other varieties, or is it a standard level? Because maybe if there is more glutathione, it can also be good for hygiene purpose with less oxidation of the product. So this is one of the points that I was asking to myself also. So I don't know if Professor Enoki is not there. I don't know. Do you know, Torhu, how much glutathione there is?
I've ever measured the glutathione, but I think I didn't measure the Koshu. Okay. So I have no data now. I'm sorry.
No, okay. Okay, is there some other question or comment? I don't see a comment or a person asking questions. So in this case, well, for me, I have no more questions. So, and so I think it's probably time to thank all the panelists, all the professors that were sharing the different aspects of Japanese viticulture in Japanese viticulture. And so thank you to all of you for your nice presentation and the time of preparation and question. And so we can continue this afternoon at 2 o'clock Paris time, of course. And we will have the second session that will be focused on the viticultural approaches and solutions. The persons that will be presenting will be Vittorino Novello from Torino University. We will have also a presentation of Laurent Torregrosa and David Le Courriot and Dr. Drive from CSIRO. And then we will have also a presentation of our colleague Antonio Guaca concerning the vineyard evolution with the Med-Gold program, and the discussion and question as we did just now. And so I thank all of you for your participation, and I say to you, see you at 2 o'clock for the second session. Thank you very much.